In this video, I want to tell you about the visual system, in particular the mammalian visual system. Why? Well, the goal is to learn about the mammalian visual system, which is the basis for many neural network vision systems or models. So I want to show you something about your visual system, which is what many scientists and neuroscientists and computer scientists have used to design better neural network models for vision type input. Okay, first of all, most of the networks that we've looked at, in fact, pretty much all the networks that we've looked at in this course, have assumed that between different layers, it's all-to-all connectivity. So every neuron in one layer is connected to every other neuron in the adjacent layer. Okay, but that's not the way your brain is wired. In fact, you've got about 86 billion neurons in your brain. About 16 billion of them are in the cortex, which is the outer shell that looks all noodly. If they were all connected to each other, all those connections just wouldn't fit in your brain. So your brain is actually a very sparsely connected neural network. Okay, so as an example of the wiring in your brain and some of the wiring strategies and design strategies, architectures, let's look at some fascinating features of your visual system. So first of all, it's layered. So although the details are more complicated, your visual system is roughly arranged into a hierarchy of layers. So here I'm showing this graphic from Jonas Kubilius. And you can see the information goes into your eye and activates photoreceptors on the retina. And now the retina is actually has a bunch of neurons in it. So it's the only part of your brain that's actually outside of your outside of the brain case, I guess you could say. But your retina is an extension of your brain, and there are neurons that go all the way through the optic nerve to the lateral geniculate nucleus. I don't expect you to memorize all these names, but just for those who are interested. LGN, lateral geniculate nucleus, and then into the back of your brain where most of the visual processing starts. And then there goes through V1, V2, excuse me, then I don't know where V3 is, but then to V4, and eventually to a region called IT, which I'll describe a bit later on. So you could, again, skipping a lot of the details, it's sort of like you've got a multi-layer neural network where you've got your input over here, you go through the retina, and then the lateral geniculate nucleus, then to V1 here, V1 here, and then to V2, V4, and then eventually to IT. So these networks are showing these sort of feedback connections. We're going to ignore those for now. In fact, there are also feedback connections here between layers. We're going to ignore those as well, as many neuroscientists do. In my research, I actually do deal with those feedback connections, feed forward and feedback connections. But your visual system is roughly layered, hierarchical. And at each layer in the hierarchy, you can imagine that the information, the visual information that's gone in through your eyeball gets more and more abstract as you go up each layer. At the very lower layers of the retina, certainly, things are basically just pixels. The image is represented by a bunch of photoreceptors that are essentially pixels. Your eyeball, each eyeball is about a 1 megapixel camera, if you like, because the optic nerve going into the LGN has about 1 million ganglion cells or sort of neurons. So it's conveying about 1 million pixels worth of neurons. Then you get into lateral geniculate nucleus, and then V1, and then V2, and as you go up and up the layers, it dissects the visual input into different representations, which are typically more useful for representing the types of things that you will see. Because ultimately, in IT, which is the inferior temporal region of your brain, temporal lobe is this sort of this thing hanging off the side here. The inferior temporal region contains a bunch of neurons. It's also called the fusiform gyrus, or the fusiform face area, maybe you've heard of, where neurons seem to respond to specific objects. Like a pair of scissors, there will be a set of neurons that only get excited when you're looking at scissors. Scissors could be anywhere in your visual field, but for some reason those neurons care most about scissors. You might have heard of the grandmother cell or the Jennifer Aniston neuron. They're referring to neurons in the IT area, where they're specific to objects. Somehow your visual system, through layers, has abstracted out particular patterns of input that fit these complex objects, like people and tools and whatever. It's layers. Your visual system is topological. Now let me describe what that means. Topology, or topological in general, means that there's a continuous mapping, continuous spatial mapping between two different spaces. So in terms of your visual system, it means neurons close to each other in the primary visual cortex process parts of the visual scene that are close to each other. In other words, nearby neurons in the back of your brain, in your visual cortex, neurons that are close to each other in the cortex are processing parts of the visual scene that are close together. So let me describe that in a number of different ways. First of all, let's zoom in on this picture here. So here you have the visual scene out here, so the world that you're looking at. Now, let's split the world into a bunch of, I'm going to put a bit of structure in the world here. So we've got, let's start with the, what do I want here? Okay, red. I'm going to kind of put a rainbow of inputs here. Okay, so we have a whole bunch of different colored inputs. So that's just the world, and I've just colored it spatially. Now, you can see both eyeballs feed into both sides of your brain, but your brain is split into two different hemispheres, split along this line here. Now, half of each visual field, or half of the visual field, goes to one side and half goes to the other side. This brings up all sorts of really interesting things, neuroscientific phenomena, but I won't go into right now, but it's really quite interesting. So the right half of the visual hemifield, which I've shown here as the red, orange, and yellow part, will end up in the left part of the brain. So you can see these sort of burgundy lines that feed through here and then end up in the left side of the brain. And in particular, the ordering is such that on the center of your vision, this yellow here, ends up in here. Then you get this part out here, and then the peripheral part ends up out here. And I can do the same thing on the other side. Green in the sort of center of your vision. Blue, I mean, the colors are just color coding, of course. It doesn't say anything about the reality of what you're looking at. But this just shows how the visual scene is spatially arranged. It is also spatially arranged on your cortex. And half of each eyeball goes to the one hemisphere, and the other half goes to the opposite hemisphere. But both eyeballs project to both hemispheres. Okay, so now looking at the details of this, what we're showing here is kind of a part of the visual cortex, sort of this kind of opened up so you can like see inside. Whoops. So you can see inside the cortex. So I've drawn this dot here on this visual field, sort of what the person is looking at. In fact, it's drawn here in the visual field. And then now let me color code some of the pieces here. So let me do the first 10 degrees in yellow. Let me put it above here. 10 degrees in yellow. Then I'll do the next 10 degrees in orange. Oops, it should be like that actually. And then the final 10 degrees I'll do in red. Or next 20 degrees in red. Okay. So how does this color get depicted on the cortex? Now, first thing that you can see over here is that the different spatial regions, starting from the center out, like almost like we have here left is like a dartboard. The center region has a much higher density of photoreceptors. So proportionally speaking, it's almost like a fisheye lens where there are more neurons processing this central region than the peripheral region. Okay, so that's what we're seeing over here is the stretching of the space to give more resources, more visual computing resources to processing the center of your vision. You'll notice if you're reading a book, you can only really read the words that you're looking at. If you hold a book out at arm's length and you try to read it, you can. But if you kind of shift it away and you look like to the side of the book, you can't read it anymore because that part of your visual scene in the periphery is lower resolution, so you can't make out the words. If the words are really big, then you can read them because you don't need a high resolution high resolution camera to make out big words. I mean big letters, not big words like photosynthesis. Okay, so more resources concentrated on the center of your vision, fewer resources focused are concentrated on the periphery. And you can see that here because let's go back to yellow here. The 10 degrees goes all the way out here. Oh my goodness. All the way out here. That's all just from that little region of the visual scene. The next 10 degrees is here, and then the final 10 degrees, or 20 degrees I guess, is out here. So you can see the peripheral gets less real estate in your cortex. And actually drawing it on the cortex, here's the 20 to 40, here's the 10 to 20, and then here's the 0 to 10 arranged on your cortex. And that dot appears at about 18 degrees or something, so that would be over around here. So that dot would excite a bunch of neurons around where that red dot is in the cortex. Okay, so if you like, it's almost like the visual scene is projecting like a warped video screen on your cortex, and in the sense that neurons are firing. There are experiments where neurons that are firing light up, and so you can actually see the neurons in the cortex lighting up according to the scene that you're looking at. The original experiment that established this was based on a cat looking at a particular scene, and they had treated the cat with a chemical, injected a chemicals that would become concentrated in the neurons that were firing more. And they, sacrifice the cat, take its visual cortex out, flatten it out, and then look for the presence of that chemical. And it was in the shape of the scene that the cat was looking at. Okay, so I hope I've described this story fairly reasonably well. The point is that it's topological. So each neuron in V1 is only activated by a small patch in the visual field. Let me do a quick joke now. According to the Institute for Incomplete Information, 9 out of 10 people Okay, so here's a visual field. Now let's look at a particular little region of visual field here, let's say. So that region excites a neuron in the cortex. I'll just sort of draw some neuron here, an axon. So this little visual field, or that neuron, is only going to be interested in that part of the visual field. Okay, and a different neuron would be interested in a different part of the visual field, and so on. So you've got millions and millions of neurons in your V1 that are each sort of each assigned their own region of the visual scene to process. Okay, conversely, so each neuron is only excited by a small region of the visual field. Conversely, each patch in the visual field only excites a small neighborhood of neurons in the primary visual cortex. Okay, so the sort of mapping goes both ways, and I'll allude to that a bit more soon. This topological mapping in the visual system has a name. It's called a retinotopic mapping. Retino because it's from the retina. So the visual scene that sort of spatially lands on the retina maintains a lot of its spatial information, even in the neural resources assigned to it in the cortex. Moreover, neurons in V1 project to the next layer, V2, and again, the connections are retinotopically local. So let's look at a single neuron in V1 and look at sort of the cascade of neurons in V2 and subsequent hierarchical layers in the visual system that are affected by it. So this neuron in V1 is going to be in the footprint or the receptive field of a little neighborhood of neurons in V2. So this is V1 here. Let's call this V2. So V1 will project to a neighborhood in V2. Each neuron projects to a neighborhood in V2. Now this neighborhood projects to each neuron in that neighborhood projects to a neighborhood in the next layer. Or if you like, the entire neighborhood projects to an even bigger neighborhood in the next layer. And so on. You go up to the next layer, the neighborhood gets even bigger. So every neuron in this region over here has input ultimately from this neuron way over here on the left. So information is going this way. We also got a different neuron here. So it's a different neuron in V1, so representing a different part of the visual field, but it projects to a different region in V2, which again projects to a sort of bigger footprint in the next layer. And you keep working away through the visual system, its footprint gets bigger and bigger, and you can see I've drawn them overlapping quite a bit. So sort of the future footprint of each of those two areas of the visual system overlap quite a bit in these upper layers, until eventually you get to the IT, which stands for inferior temporal. Again, I don't really care if you know or don't know these. In which case, both all the visual scene influences all neurons in IT, and that's why you get neurons that are specific to particular objects. The pair of scissors can be anywhere in the visual scene, and it will excite the same scissors set of neurons, right? There might be a small set of neurons that really just care about scissors. Okay, so the footprint of a region in the visual field gets larger as the data progresses up the layers. And the neurons in the last layer are influenced by the entire visual field. Okay, so that's the retinotopic or topological nature of your visual system. And finally, your visual system is a bit like a filter bank, at least at the lower levels. It's easier to identify the filter bank aspect of the visual system. So let me describe what that means. In the lower levels of your visual system, the neurons, you can talk about you can look at a neuron in V1, primary visual, the first sort of cortical processing of your visual scene. Look at a neuron in V1, and then look at what in the visual scene excites that neuron, and what most things won't excite it. But the question is, what particular visual configuration excites that neuron the most? So what I'm showing here is a bunch of receptive fields. So receptive field is the term for the configuration of the image that excites a particular neuron. So each of these little squares here, let's look at this one, corresponds to one neuron in V1. So a particular neuron likes that particular template shape. It likes kind of it's sort of sensitive to information like an edge that's on like this, I don't know, whatever angle that is, sort of 115 or 130 degrees, something like that. And different neurons are sensitive to different orientations. And in fact, V1, a lot of the neurons seem to be little edge detectors. So you can see these are kind of like interested in edges, edges that are oriented at different angles. So that one's like a 45-degree edge detector, if you like. So these are found in mammalian visual systems, and this is a monkey visual system, a macaque. This thing on the right, the sail net, I don't know much about this neural network. It's some sort of spiking, sparse neural network thing. The citation's down here, but I'm just using the image to show that many neural networks that are trained using some of the same strategies that the mammalian visual system uses end up with receptive fields that look like mammalian visual receptive fields. So this is from a neural net. You see a lot of them are basically like little edge detectors. But I find that really fascinating, that the first sort of layers of your visual system essentially break up the visual scene into a bunch of little edges here and there, or oriented edges. And the next layer of your visual system, V2 I guess, can combine those little edge pieces together and start making slightly extended edges. The next layer and the next layer will build these like big curves and things like that. And the next layer, until you get to the IT, which essentially is an abstract representation, if you like, a latent representation of objects that you find have utility, like scissors and pencils and equations and symbols and people and desks, computers. So I wanted to go through the visual system and describe some of the features, because you'll see that many of the neural networks that model visual systems or model vision systems have many of the same architectural designs.